All right, it's already 12:30. Time flies, so let's get going so we can be done eventually. So today we're gonna continue going over Chapter 6. Hopefully we'll finish Chapter 6. The quiz for Chapter 6 is posted in Brightspace. It is due a week from today, so next Thursday night. So make sure you do that quiz. It's quiz number 5. It's about Chapter 6, in case that's confusing for anyone. So here's where we ended on Monday, just talking about dihedral angles and Ramachandran plots. So we will continue from there. So let me make sure I'm recording. Yeah, okay. So let's go on to, you have a question? Yeah. Okay. Yes. Yes.
Is that on Brightspace?
Yeah, I'll post the scores on Brightspace. I will be honest, I'm not completely done grading it, but I'm hoping to be done grading either tonight or tomorrow with most of the exams, and I can give out scores. There's still a couple people who have to do makeup, so I won't post like the answer key yet, but I will once everyone's done taking it. Yeah. Yep. Any other questions before we get started? Okay, so yeah, so we ended on Monday talking about dihedral angles. Today we'll do a couple more questions. So we'll start with a Ramachandran plot for a real protein. So for this question, we have plots that have phi and psi angles, and they look a little bit different than the plots we saw like from the textbook. So from the textbook, the favorable and preferred dihedral angles are labeled in blue and green. Here, our favorable angles are labeled in red. It's hard to see on the screen. I apologize. And the acceptable angles are in yellow. And then everything else is like a white background. Those are unfavorable. And then the actual angles that are measured for a protein are labeled as these little black dots. So everybody take a couple of minutes and think about each of these two plots that are labeled A and C. What types of secondary structures are happening in each of those plots? All right, so hopefully everybody's had time to think about these two structures. So for the structure labeled A, what kinds of secondary structures would you expect to see based on those dihedral angles? Yeah.
Beta sheets, coils, and inverted alpha helices. I'm very confused at what's at like 0, -90 and above.
0, -90, like
0 on the y-axis.
Oh, 0.
Like, I guess -120 to like -75 or something. Things in that area. Because it's really, that area would be for beta sheets, but it's like not supposed to go up that high.
Like around here? Is that what you mean? Sorry.
0 on the y-axis.
Okay.
and from -120 to like -170.
Oh, like around here.
Yeah.
Okay.
So the bottom
Yeah.
of that will be beta sheets, but the above part shouldn't be there.
Right. Yeah. So so that area is kind of an extended version of where you'd find the alpha helices, the right-handed alpha helices. Which so on the plot from the book, those are kind of like here. So the way that this one just sort of like extends that out a little bit further. So like you can consider that as like part of the alpha helix region, I would say. So yeah, that's one thing you'd see in the structure is a bunch of alpha helices, right-handed ones, which are circled over there. The other thing you see a lot of, like you said, are beta sheets, which are up here, and that also wraps around to down there. You don't really see like distinctive marks for like the parallel and anti-parallel beta sheets, but you definitely see a whole bunch of both. Yeah. Yes, and then exactly. So over here we would have the left-handed alpha helices. Yeah, so this protein structure has a little bit of everything. Yeah.
Would you ask us, like, on the quiz or exam to identify whether it's right or left by looking at it? Like, do we have to know that rule about the thumb and stuff?
Oh, so I wouldn't like give you a picture of an alpha helix and tell me is this right or left-handed. That's more for you to tell the difference. Oh, and also speaking of that, I think I told you the incorrect way to figure it out last time. So your thumb is pointing towards the C terminus, and then the way your fingers wrap tells you if it's right or left-handed. So just to be clear about that from Monday. But from a Ramachandran plot, you should be able to tell if they're right or left-handed based on where they're located on the plot.
Okay, but not by looking.
But yeah, I don't think I'm gonna just give you like, here's a picture of an alpha helix. It's more just to know like about it. Okay, so that's our protein A. What about our protein C? What kind of secondary structures are we seeing there? Yeah. Yeah, exactly. So we see almost all in this region over here, which are our right-handed alpha helices. And those are the more common ones anyway. In general, you see more right-handed ones than left-handed ones, but it depends on the individual protein, of course. So, so this is, given a protein that has angles, you can sort of predict the secondary structures. And then these proteins that we're looking at plots for right now are actually two different proteins that we might be familiar with from SARS-CoV-2. So the one labeled B over here, that's the spike protein. And that is corresponding with our plot number A. So in plot A, we were saying we expect to see a lot of beta sheets and a lot of alpha helices, kind of a mix of everything. And you can see in this picture of the spike protein, which again is a little bit hard to see on the screen up there, but you see in green a lot of beta sheets. You see in that kind of orangey color a lot of alpha helices, and then super hard to see here, but in kind of that purple that blends in with the background, you also have kind of random loops and coils and stuff. So it's kind of a mixture of everything, which makes sense given what the Ramachandran plot looks like, because it shows us we have a mixture of everything. Yeah.
Are there random, like, single dots in the unfavorable locations of do those correspond to loops and terms?
Potentially, yeah. So a lot of especially within loops and more unstructured regions, there isn't a fixed angle necessarily because there's a lot of movement. So this plot, I would have to go and check the actual paper where it's from, but I believe it's like a predicted angle based on like, they figured out the order of amino acids. They used a computer program to predict the structure. And then from the predicted structure, they then generated this plot based on the predicted angles from that predicted structure. So in real life, you probably wouldn't see them hanging out in like these areas, at least not for a very long time. But like if you just take a very quick snapshot, they might briefly be in that unfavorable location. Yeah. So that brings us to our other protein, protein C, which we just said was almost all alpha helices. And so protein C corresponds to protein D in this picture, which is our envelope protein, which, as you can see from the picture, is mostly alpha helices. So hopefully that makes sense. And so like Ramachandran plots can just be a really helpful tool when you're looking at new proteins, just to see sort of like if you're trying to predict the structure of a protein where you know maybe the sequence, but you don't know what shape it takes on. It can be handy to look at a plot like this to see, is this likely to happen? Is it likely to have this shape versus a different shape? So that brings us to protein folding. So we talked a lot about secondary structures so far, but how do we form secondary structures out of our primary structure? The protein has to fold. So here is this protein folding energy diagram. So I'd like everyone to take a few minutes and label these kind of 6 general areas of where the protein would be, or like where, at what like location on this diagram each of these energy levels or protein folding states would be. And I'll give everybody maybe 5 minutes for that. Yeah, that was there when I got here, Hannah. It's Friday the 13th. We got spiders now. It's Halloween. Yeah, okay, so let's start talking about this protein folding energy diagram. So who can tell me where the highest energy level is located on this diagram? Yeah. Yeah, exactly. So if you think about this as kind of a graph, as you go up, you have energy increasing as you go up. So the highest energy areas of this graph are kind of anywhere at the top. So between here and here, we have our highest energy. So I'll label that A. Where would we have the largest number of potential structures? Yeah. Also at the top. Yeah. So at the top of this diagram, yeah, I don't want to give away what's happening, but yeah, they have a large number of potential structures for your protein, and it's also the highest energy level, which brings us to C, which is where is this going to have the most stable structure? Yeah. The lowest point, exactly. Yeah, so C is down here. We have our most stable structure, and that is sort of goes inversely with B, right? So where we have the largest number of structures, that's where it's kind of the most unstable, towards the top. Towards the bottom, where we have that funnel kind of coming to a point, that's our most stable point, where we have the least number of potential structures. That's where like the protein has found its ideal folded state. So it's very stable, and there's like only one potential or very few potential conformations. Yeah.
So when I was like learning it, I made a hard time with structures at like very good and C as well. Because I thought that largest number of structures meant like the most number of like beta sheets, alpha beta, like that's the kind
Ah,
of all folded.
okay.
So I guess like how would we distinguish?
Okay, yeah. So I guess that's that comes in where with the question wording. So yeah, you are, I guess, right that you'd have like the most amount of structure, but when it's asking the number of structures, yeah, that I guess that wording is just really confusing. So yeah, what we're getting at with the protein folding diagram is protein folding as a whole. So when you're asking for if you're looking at a diagram like this, when you're talking about structures, you're talking about potential fully folded protein structures, or potential structures for that protein to take on, whatever shape that is, like as a whole protein, as opposed to like the individual parts of the protein that might take on different secondary structures.
So like there's only one. So the final combination, there's only one for every protein because that's the right function that it will like out. But the diagram itself can change based on how it gets to that final folded state.
Yeah, so like you can kind of imagine it as like a process that's happening like over time. So like at the top of the diagram, you have, I guess I would give away some of the other answers. Let's go over the other answers first because I don't want to like give away the other half of the question, and then we'll talk about it. Yeah. Okay. So who can tell me for D, where is the unfolded polypeptide? Yeah. Exactly. Yeah. So we have our unfolded polypeptide, which is the largest number of potential structures because it's completely unfolded. It can do anything at this point. So for part E, where are the partially folded structures? Yeah. Yeah, so like anywhere in the middle here that's like not the top or not the bottom, here are all of our partially folded structures. And so, yeah, so then where would we have the highest entropy? Yeah. On the right. So you mean like on the right towards the top? Or yeah. So yeah, it would be the right or the left. So just anywhere along the top, just like with these other ones. So the highest entropy means essentially the most disordered, which goes along with when it's unfolded because there's no structure to it. It's just unfolded. You have a lot of potential structures it can form, and it's high energy. So yeah, so if you think about this diagram as a whole, you can kind of imagine it as like a journey that this protein can take, right? So at the top, it's like a fresh new protein. It's unfolded. It has its whole life ahead of it. It doesn't know what it wants to be, and then it's going through all these changes. It's trying to fold because it has all this high energy. It's like it's unhappy. It really wants to be happy by lowering its energy. So it tries to fold at all these different angles, and you can see like these peaks and valleys as we're like folding and trying out new different shapes. And then eventually it should reach a point where it's its most possible stable self, and that's where it's the most happy. It's the lowest energy. It's the most stable. It really doesn't, once it reaches that point, it doesn't want to leave unless you do something to change its environment to make it unhappy again. Yeah.
Why is it that we're looking at a V shape rather than a one-way street?
Yeah. So you can sort of think of it maybe as like folding and then like one side for folding and one side for unfolding in a way. Like on the way in and then on the way back out. If that kind of helps. Or just like, or you can imagine it as kind of like a funnel where like the top is the widest and you have the most possibility, and then it gets like narrower and narrower towards the bottom. I don't know why they decided that this is the convention for drawing these types of structures. Sorry, yeah, but that's just how you see them. Yeah, there must be a reason. I can look that up, but yeah, I do not know why. Yeah, yeah. Yeah, so the highest entropy is at the top. So, oh, like does the x-axis correlate with entropy? Is that what you're asking? Yeah, yeah, that's like one reason. Yeah, so so I think it's more helpful to think of it being wider at the top because it has the most number of potential structures. So like if you can imagine like all the potential shapes the protein can take on, it needs like more space to like fit into this area because there's a lot more potential shapes up there. And as you go down this funnel, there's a lot fewer potential shapes it could take when it's at this point because it has like some structures formed, but not all of them. Or like when it's down here, it's like eliminated most of the higher energy structures. Yeah.
So, I'm a bit confused about like entropy because like having high entropy is a favorable condition, right?
Yeah. So that was something I wanted to talk about too. So yeah, so protein folding, as you just pointed out, is entropically not favorable, right? When a protein is folded, it is at its most orderly state. So, protein folding has a positive delta S, as we recall from our chapter 1, I think it was. But the good thing about protein folded is that it is enthalpically favorable. It has a negative delta H. It's exothermic. So, sorry if you can see that. So you have this balance, right? You have, and if you remember our Gibbs free energy equation, so you have G equals delta H minus T delta S. And we want our delta G to be negative for this reaction to be spontaneous, right? So now you can figure out sort of what conditions the protein is going to fold in based on temperature. So if you have a negative delta H, you want to minimize the contribution of that positive delta S. So at low temperatures, this is going to be favorable. So proteins are going to want to fold at low temperatures. And then on the other hand, like because you have a positive delta S, so like if you have a high temperature, that's going to make that term more favorable. And so you'll get an unfolding of proteins. Yeah. Did I say that backwards?
Positive delta S, negative delta H is always favorable.
Yeah. Oh, sorry. I lied to you. It has a negative delta S. Okay. I confused myself. Yeah. Or I said one thing and wrote the other thing, and then I confused myself.
Oh.
Yeah, so we have decreasing entropy when proteins fold. Sorry, I said that and then wrote the opposite of that. So now what I said should make more sense. So if you have a negative delta H and a negative delta S, to minimize the contribution of the negative delta S, you want a low temperature. And so proteins are going to want to fold when they're colder. When they start to warm up, that negative delta S term becomes more important in this equation because the temperature is larger. And you don't want to make that term too large because then delta G will go positive. Your protein will unfold. So that should make sense. Yeah.
Is there any example optimal conditions for in that optimal well not pH but in this case optimal temperature for protein folding from like this system? Or is that
I'm not sure exactly what you mean by like the optimal.
there's like a range that proteins can fold and then like outside of that range they either premature or they just don't fold.
Yeah.
So is this like how you get the optimal temperature?
So
This calculation?
So yeah, I guess you could for an individual protein determine like the exact delta H and delta S. Yeah, I think there are experimental ways to do that, and I'm like, I know you can do like calorimetry to calculate H and stuff. But yeah, I mean that's where you get that idea from, right? Like every biochemical process is ultimately has to be thermodynamically stable in some way, and so yeah, so proteins are only going to fold if it's favorable for them to do that, and they will unfold if you're outside of the range of those conditions. And so like temperature is included in that. There are also things like pH that you mentioned that will also affect whether or not the protein wants to fold. Also like what solvent it's in is going to affect whether it wants to fold. Yeah, there's a lot of stuff will affect it. Yeah. Okay. Any questions about this? Okay, so let's now talk about protein folding and how it's reversible. So we had this diagram in the lecture slides, and so we talked about there are some reagents that you can use to unfold and refold proteins. So everybody take a couple minutes and think about how this experiment that we're doing here is showing us how protein folding is reversible.
Oh, sorry. Where is this slide?
Like from the lecture.
It's in the lecture, but it's not in the questions.
Oh, it's not? That's
Not on
weird.
mine.
That's interesting. Okay, well, I don't know how that happened. Sorry. Alright, so here is our diagram. So at the top of our diagram, we have an example of a protein in its properly folded state. So what happens after that first arrow? What happened to the protein? Yeah, it is denatured. Yeah, so what reagents could you add to do that to the protein? Yeah, exactly. So we added urea and BME. So let's get more specific. So what is the BME doing to the protein? Yeah. Yep. Yeah. So BME is reducing disulfide bonds. And we see that illustrated in this picture because all those yellow disulfide bonds are now thiol groups just hanging out. And what is the urea doing? Yeah. Exactly. Yeah. So the urea is just like generally unfolding by disrupting the other interactions in the protein. And so by using the two of them together, now we have a protein that is completely unfolded. So, yeah, so then the next arrow in our pathway here, we have an oxidation occurring. So how do we make that happen? How do we refold the protein? Yeah. Yeah, and so, and you'll notice this forks left and right. So if you remove both of them at the same time, would you go to the left or the right? To the right, yeah. So if you go to the right, we remove both urea and BME. If you remove them together, the protein is going to be able to take on its native state. It's going to reform the disulfide bonds all in the same places as the original. So if you look at the original protein and you look at this refolded protein, they're the same because that's how the protein wants to be. That's where it's happiest. How can we force the fork to go the other way, to the left? Yeah. So if you want to remove one of them at a time, but you can see in this diagram on the left, we've reformed a certain type of bond. What type of bond did we reform there? Yeah, the disulfide bond. So which reagent did we remove first? BME. Yeah. So now the protein is in this state where it's reformed disulfide bonds. And it's a little sort of hard to tell by looking at it, but these disulfide bonds are not the same ones that were in the original protein. Now we have different thiol groups bonding with other thiol groups instead of the original pairs that we had that formed those original disulfide bonds. So then our next arrow to get us to the bottom of this diagram, now we have the protein in this folded state. So what reagent did we remove to get there? Yeah, urea. Yeah. So now we have what we call this scrambled protein. So we call this protein scrambled because its disulfide bonds are all scrambled, and so that gives it a structure that is not its original native structure. It has this other structure where it's less happy than it originally was. It's a little bit probably less stable. This protein might not perform the same function that the original protein had because the location, like its tertiary structure and its secondary structure, are all mixed up. So how can we then take this scrambled protein on the bottom and bring it back to its happy native state? Our last arrow. Yeah. Yeah, so you would add just a small amount of BME. And so by doing that, that little bit of BME is enough to break those disulfide bonds that are less favorable. But there's not so much BME in there that the bonds are not able to reoxidize on their own, given that there's sort of favorable like oxidation conditions in the buffer that it's in, like in the rest of the environment where the protein is. And so, yeah.
Are the intermediate states, so like before you remove the urea as well, can you also call that a scramble? Because you know, only when both of them are removed, but not fully like actually called scrambled.
Right. I would say you could call either one scrambled. Yeah, I mean, I guess like this one over here is sort of scrambled, but it's still partially unfolded. So there's more to it than just being scrambled, but that's part of it for sure. Yeah.
And then also, how would this change when you remove urea?
So I think in that case you would get probably correct formation of disulfide bonds, because when you remove the urea, the protein can fold and form the angles that it wants. It just can't connect together some of those thiol groups. And then if you remove the BME afterwards, theoretically, like once it's refolded, the thiol groups are in the right locations to just form disulfide bonds with their correct partners, because they would be near each other in space if the protein is folded the way it wants to be.
So would you like kind of go down the right side, but with an extra step in the middle?
Yeah, I would say you could do that. Yeah. 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 Yeah.
If you remove both urea and BME at the same time, you will always get the right folding.
Theoretically, yeah. In practice, maybe some proteins are pickier than other proteins about what conditions they want to fold in. I will say, as someone who makes a lot of proteins in the lab, they don't always want to fold, right? They get cranky, but in theory, this is what should happen. Yeah. Any more questions about protein folding, how to unfold and refold proteins, stuff like that? All right, so let's move on to our structure determination techniques. So we discussed 3 main techniques in this part of the chapter 6. So we talked about circular dichroism, or CD, nuclear magnetic resonance, or NMR, and X-ray crystallography. So everybody take a few minutes and think about all of these statements and which techniques apply to which statement. Or you can just take like one technique at a time and think about which statements apply to it, whatever is easier for you to think about. And I'll give everybody maybe 5 minutes. So let's talk about each of our structure determination techniques and think about what statements apply to which one. So let's start with circular dichroism, or CD. So who can tell me which statements apply to CD spectra? Or a statement. You don't have to tell me all of them. Yeah. 3. Yeah. So circular dichroism definitely measures the absorption of light by chiral molecules. Excellent. What else does CD do? Yeah. 6. Yep. It monitors changes in the structure in real time. So CD spectra are fairly quick to acquire. So you can, if you have something happening to your protein, maybe it's binding to something and its shape is changing, or some other, maybe you're changing its environment or something else. CD spectra are a really simple, straightforward way that's quick, where you can, every few minutes, you can take another spectra and see if you have changes in structure. So, yeah. So what level of structure are we looking at with circular dichroism? Yeah. Secondary. Yep. So yeah, it can tell us how much alpha helix versus beta sheet versus random foil that we have in our protein. And those are things that might change if the protein's changing its shape for whatever reason. So, okay, so let's talk about nuclear magnetic resonance, or NMR. So can somebody tell me a statement that applies to NMR? Yeah. 7. Yep. So nuclear magnetic resonance, that's sort of how it's defined. We're probing the decay of aligned nuclear spins. So we apply this magnetic field, all of the spins align, and then as they decay over time, we see differences in decay that, after doing a lot of math, we can turn into a spectrum that can tell us a lot about the structure of a molecule. What else can NMR tell us? Yeah. 5. It actually does not produce an electron density map. That is tricky, though. So NMR tells us more about protons. It doesn't tell us a lot about electrons. Tricky, though. Yeah. Anybody have a different one? Yeah. 2. Yeah. So NMR can provide a collection of 3D structures in solution. So yeah, so we've talked a lot about how some secondary and tertiary structures are relatively fixed. So things like beta sheets and alpha helices, they're not going to move around a lot. They have fairly fixed angles. They're going to look the same when a protein is in solution. But there are parts of a protein that are more flexible, so like loops and more unstructured regions. So if you have a whole bunch of different individual protein molecules in solution, they will be, every individual protein molecule will be a little bit different from its neighbors, and they'll also change over time. There's going to be, wiggling and vibration and other small movements in those unstructured regions. And so because NMR is measured by looking at proteins that are in solution, NMR actually can provide you with a whole collection of all the different possible structures you would see with that protein. Because NMR is telling us the relative distance in space that the protons have from each other. So it's sort of an average of all of those 3D structures that we might get. So yeah, so that brings us to X-ray crystallography. So who can tell me, I guess it's sort of the leftover ones, but who can tell me what statements apply to X-ray crystallography? Yeah. 1, 4, and 5. Yeah, those are the leftover ones, right? So X-ray crystallography gives a single 3D structure. So that comes of, again, how this technique works. So X-ray crystallography works by very carefully and painstakingly making a crystal structure out of a protein. And that crystal structure is going to be that one most stable, ideal shape and structure for that protein. And you'll have a whole bunch of protein molecules that all have the exact same 3D structure, and that's why they'll form a crystal, because it's repetitive. It wouldn't be able to form a crystal if you didn't have those like repetitive shapes, essentially. So that's number 1. So number 4 collects an X-ray diffraction pattern, kind of in the name, right? So X-ray crystallography, we're shooting X-rays through this crystallized protein. It's going to bounce off electrons, and X-rays will diffract and make this pattern that you can then deconvolute using a lot of math to then come up with a structure of your protein. And that goes along with number 5, which is we're producing an electron density map. So the X-rays are diffracting. They're showing us where there's more electron density, where there's less electron density. So we can look really, depending on the resolution, you can see really clearly like where bonds are, and you can get a very detailed structure just based on if you get really good quality X-ray crystallography. Yeah.
Is NOSI considered a protein density map?
I don't know if you would call it proton density necessarily. It's measuring the relative distances of protons to one another. So if they're within a certain distance of one another, you will get a peak at the intersection of those two protons. So it's not necessarily about density, it's about interactions of different parts of the structure with each other. Yeah. That's a good question. I know for sure it applies to NOSI. I'm not as familiar with the other ones. I know there are a lot of other ones. Off the top of my head, I don't have details on some of the other ones, so I'm not sure. I would guess probably, but not sure. Yeah.
So it's like just like conceptual to understand so when two photons are within that distance, then they form a peak. That interaction results in depicts as a peak in the NMR.
Exactly. Yeah. Yeah. Oh, and then before we move on from this, I just wanted to make sure everyone is clear. So for circular dichroism, we're looking at secondary structures only. For NMR and for X-ray crystallography, we're looking at what level of structure? Yeah. Yeah, exactly. We're looking at tertiary and quaternary. Can you choose? Like which level? Not necessarily. I mean, I guess if you had a protein that had multiple subunits and you didn't want to look at quaternary structure, you could probably figure out a technique to separate out those subunits and purify them away from each other and look at each subunit separately. But yeah, in general, you wouldn't be able to tell the difference necessarily between tertiary and quaternary. Especially for NMR, you're just looking to see are the protons near each other or not. You don't know if they're on the same polypeptide chain, if there is more than one polypeptide chain.
Wouldn't that just mean that you're only looking at quaternary?
Well, not all proteins have quaternary structure. So if a protein only has one polypeptide chain, you're looking at the highest level you can go is tertiary.
Okay. I would think at that point then that tertiary is quaternary.
Yeah, I mean, in some way you can think of it as the highest possible level of structure. That's what you're looking at. If you want just one definition or one answer. Yeah. Yeah. So I mean, you can think of it sort of in contrast to X-ray crystallography. So with X-ray crystallography, you have very painstakingly made a crystal out of the protein. So all the protein molecules in that crystal have the same exact bond angles and structure and precise shape and conformation. When you're doing NMR experiments, your protein is in solution. So when your protein's in solution, it has a little bit of freedom of movement. So if you have a disordered region of your protein that doesn't have exactly defined dihedral angles, you might have some loops that go this way, some loops that are going that way, or like 5 minutes later, it's moving back and forth. And you have a whole bunch of protein molecules in your solution. It's all the same primary structure, probably all the same secondary structure, but you might see small differences in tertiary and quaternary structure for NMR just because your protein is in solution. So it's not to do with the NMR technique itself, it's mostly just because your protein is in solution, and so it doesn't have only one exact precise structure.
Okay. And then when most confused about it, then what's different between nosy and then just regular if they both possibly should amplify angstrom?
Right, so NMR is a broad term, and there's a number of specific NMR techniques. So nosy is a subset of NMR. So if you think of like, I don't know, classical NMR, it's like one-dimensional. It's really good at telling you the structures of small molecules or relatively small molecules. But when you get to really big, complicated molecules like proteins that have a whole lot of atoms and a whole lot of bonds in them, 1D NMR will just look like a mess. And so that's why people have come up with these 2D techniques and other NMR techniques, including nosy, but there are a whole lot of other ones. And each one will tell you some specific detail about your macromolecule. Cool. Okay. Any other questions about these techniques in general? All right, so let's go into specifics. So first we'll do some circular dichroism. So here is a CD spectrum. Everybody take a couple minutes and decide what type of protein secondary structure we're looking at. Yeah. Yeah. All right, so who wants to tell me which kind of structure we're looking at? Yeah. Random coil. Yeah. And how did you know that?
Well, the easiest part is it doesn't have a peak that goes up before, and that's the only one that really doesn't spike up before it goes up.
Yeah. So yeah, when we're looking at our y-axis, our 0 is shown with this dotted line. So the other types of secondary structures have more stuff above the 0. Random coils don't. But usually when we want to like positively identify our 3 different potential secondary structures that CD is showing us, we actually want to look at the valleys and where they're located in the wavelength on your x-axis of the spectrum. So your random coil has that valley there around 195, 200, and so that's a good place to look because it gets a little bit. And the reason I point that out is because those are sort of easier to see, especially if you have a mixture of secondary structures. So like if you just have one, like this is all random coils, so it's pretty easy to identify. But sometimes, as you add on like mixtures, it's much easier to look at the peaks rather than the valleys rather than the peaks. And so just to refresh everyone, this is what our 3 different secondary structures look like on a CD spectrum. So this red curve here is what we saw in the last slide, where we have our little valley around 195, 198 or so. For beta sheets, that's shown in blue on here. And it has this valley over here around 215-ish. And then for alpha helices, we have these 2 valleys, one around 205 and one around 218, 220. And so those are the most obvious or easy places to look if you're trying to tell the difference between the types of curves you'll see on CD spectrum. Any questions? Yeah. Yeah.
Do you get all of them, or how do you see right now? Or would you get more of an average?
Yeah, so you would get just one curve, and it would be an average of all the structures. So like if you had some random coil and some beta sheet, you might see a curve that has like one valley there and another valley there. Like where like this would be the 195 and this would be the 215 or whatever.
So would the minima still correspond to whatever the individual ones would look like?
Yeah, so the minima are located at the same wavelength, which is why those are a good place to look, especially if you have mixture. Yeah, exactly. Yep. Yep. Yeah. No, you should know the wavelengths and the shapes of the 3 different types of curves. Yeah. Any other questions about CD spectra? So I have another question about CD spectra. So here we're doing an experiment, and we have a protein that is changing its shape. And we can tell that because we have all these different curves that we're drawing as we are changing the buffer, the environment that the protein is in, and that is causing this protein to change shape. And you know that because the curves are changing shape on your CD spectra. So everybody take a couple minutes and check out this spectrum or series of spectra and tell me what shapes we have. What do we have at the beginning? What are we turning into? And yeah, I'll give you a couple minutes for that. All right, so who wants to tell me what is going on during this experiment? Where did we start and where did we finish? Yeah. Okay, yeah, go ahead.
We're seeing a random coil progressively turn into more and more alpha helices.
Yeah, exactly. So we're starting over here with this blue one, which I don't know, the colors might be kind of hard to see, but that's where we start. That's our 100% PB. And we start at our random coil shape. And we know that because we have this nice minimum right around 195, 200 nanometers. And so over time, we have this shift, and we're shifting into our final state, which is our 10% PB and 90% TFE in that kind of bright green color, which is over here. And as you correctly identified, that is more of an alpha helix character because we have the two minima. One of them is around 208, one of them is around 220-ish. And so those exact wavelengths don't exactly line up with our ideal wavelength. There might be a little bit of mixture of beta sheets in there as well, but it's mostly alpha helix. Yeah.
What are the PB and TFE mean?
So those are the different solvents or buffers that the protein is in. So it starts out in 100% PB, where it's basically unfolded because it's just random coils. And then as they replaced the PB with TFE, the protein was able to take on its more folded shape, which is more alpha helixy. Yeah. Yeah.
Do you just put in the whole protein, or do you choose a reasonable protein to solve?
It probably depends on what information you're interested in looking for. You certainly can do it on a whole protein, but if you're only interested maybe in one particular domain of a protein, or for whatever reason you don't want the whole thing, you can also do it on a piece of a protein. It will read whatever chiral molecules you throw in there. It will tell you a wavelength.
And
Yeah.
changes based on the amount of alpha helixes you have. It's still going to follow the same wavelength.
Yeah.
Okay.
Yeah, so like the intensity along the y-axis would tell you more of the degree of it. Like if you have this very like drastic set of curves in like our example spectrum where it's very sort of steep. And here, this real life data, we have the same minima, but they're a little more shallow. So maybe it's a little less. It's not like 100% extreme alpha helix, but it's pretty close. Yeah. Yeah. Any questions about this one? Yeah.
So you would say that
Yeah, exactly. So essentially we're like watching the protein as it's folding. So it starts out random, and then we gradually start replacing PB with TFE. TFE is a type of solvent that helps the protein to fold. And so the more TFE you put in there, the more folded the protein gets.
What is PB?
I totally forgot. I do not remember what PB and TFE stand for. That's my bad. But they are different solvents or different buffers. And one of them is not favorable for letting the protein fold, and one of them is. And I apologize for forgetting what they stand for. Should have written that down. All right, so here is another example of an experiment. This one isn't really a question, it's more just to get you an idea of the types of experiments people might do using CD data to look at protein folding. And so in this particular experiment, we're looking at the spike protein from SARS-CoV-2. And I believe this paper is from fairly early in 2020, right when the pandemic was starting, and they're sort of trying to look at maybe different treatments for like when you're infected with the virus. So these people were looking at heparin, which is a drug, and seeing if it had any effect on the spike protein of SARS-CoV-2. And if you, because as you might know, the spike protein is what lets the virus attach itself to receptors on our own cells and enter our cells. So if you have enough changes in the shape of the spike protein, it might not be able to bind to our receptors on our cells, and it wouldn't be able to infect our cells and make us sick. So that's what's sort of the idea behind this paper. If you add heparin, do you get this change in the spike protein? And they measured this with CD. So they made this graph looking at different types of secondary structures. So how much sort of helix character is like a percent of how much secondary structure, how much beta sheet character, how much turn character, and how much other. So as you already saw, I don't want you to know that much detail when you're looking at CD. You just need to know alpha helix, beta sheet, or random coil. But these people have more information that they're also getting out of their spectra. And so they're basically looking at these different colors on their spectrum and on their graph. So the purple color curves and bars are when the spike protein's by itself. So it has the highest amount of these anti-parallel beta sheets when it's purple, compared to like the orange, which is when you add heparin. So you have much less beta sheet character after you add heparin, and you get more helix character when you add heparin. So so like that is clearly showing you like the spike protein is changing around its secondary structure just because you added a drug, which is pretty interesting. And then the black and the gray bars and curves are sort of what's predicted versus what they actually measured, or the difference between what they actually measured and what they predicted. And so, yeah, you don't need to like know the details of this, but it's just another example of how a protein can change if you give it, a binding partner or a change in environment somehow. And that might have real life consequences. So let's talk about Nosy. So everybody take a couple of minutes and look at this Nosy spectrum and decide what you think the best answer is. All right, so which letter option is the best answer here? Yeah, C. Yeah. So This nosy spectrum tells us that amino acids 2 and 5 are within 5 angstroms of each other. So how do you know that? What are we looking at when we look at the spectrum? Anyone? I don't know if you're like thinking or raising your hand. Still thinking? Go for it. Right, yeah, I mean, so that's which color dots tell you that? The sort of white ones or the pink ones? Yeah, exactly. So if you look at this kind of diagonal line across our spectrum, that is telling you all the protons that you can detect in your protein. So this is obviously an extremely simplified spectrum because we only have 5 protons in there. So it's a very tiny protein. And the way you can tell if those protons are near each other in space is by looking at what you can call the cross peaks. So if you look at these pink dots, those are off the diagonal, right? And so those represent the intersection of 2 protons. So if you can drop a horizontal and a vertical line from either one of them, so from the bottom pink one, you can see that it's across from the 5 and below the 2. Same thing with the one on top, it's next to the 2 and above the 5. So both of these pink dots are telling you the same thing, which is that number 2 and number 5 are very close to each other in space because you have this intersection between them where you're getting a signal from your from the nosy process. Yeah. Sure, yeah, you could draw it if you want, but the point is like you're doing a measurement, and so like you will have a measurement of like magnetic field, or I can't even, my brain is broken, but like your measurement that you're measuring in nosy will only give you peaks at certain crossing locations.
So like the software itself gives you the ones that are really top. Like it already draws those squares for you. That's not something I manually do. Like, oh, the 2 and 5 are kind of close together. Therefore, there's some cross interaction there.
Yeah. So like the what's a good way for me to put this? So like all of these ones along the diagonal, those are all of your possible protons that are on your amino acids that are in your protein. You'll only get the pink spots that are off the diagonal if there is a closeness in space between 2 protons. So like you don't see a dot here because 1 and 3 are not close to each other. And you don't see a dot here because 5 and 4 are not close enough to each other. Okay, cool. Yeah.
Are both sides of the z-axis identical?
The z-axis. You mean like the y equals 1 line? Or, yeah, like every time you see a dot above, you see an equivalent dot below. So yeah. Yeah, it should be. I'm sure there are cases when it's not, but in theory, yes, it is. They're the same. Yeah. Any other questions about nosy? Okay, so yeah, I also just want to go for it. Yeah.
Sorry,
no, you're fine.
But in the slides,
yeah.
slide 48, the one that's like unrealistic,
yeah.
there's like you said something about, I think you were using the x-axis and you were like, oh, it's approximately, it could be like approximately 4 amino acids from each other.
Okay, that was the way you tell that is essentially because of this numbering. So like you can use a different NMR technique to tell you the order of amino acids and line those up. So like that's why like on your diagonal line you don't just have like 1, 2, 3, 4, 5. It says 1, 5, 3, 2, 4. So they also did a different probably NMR technique to figure out which protons are which, what order they're in, and that way you know how many amino acids away from each other they are. Nosy itself doesn't tell you that. Nosy tells you about the interactions between pairs of them.
And also, you're not going to be able to like make predictions of like which ones will be or which ones will hairpin, based on just like looking at this, right? All you can say is that they're close together.
Yes. Yeah.
Thank you.
Yeah. Yeah. So. Yeah, and I just want to emphasize that. Like obviously this is a very simplified spectrum, but in like in a real life one, you get enough interactions that it can give you a pretty good picture of like the shape of a protein. But of course you need a computer to help you do all that computation. That's not something you can just like look at a nosy spectrum and be like, oh yes, the spike protein. So just to be clear about that. So like if I ask you nosy questions, the plots will look more like this one, pretty straight simplified. Yeah, so now let's talk about X-ray crystallography. So which crystal structure out of this list would be the best crystal structure? Yeah. Yes. So it might be a little counterintuitive, but a smaller angstrom resolution is a higher resolution because the number that you're given is telling you, can you distinguish between things that are that far apart? So the higher resolution, the smaller that number, because if you can distinguish things that are 1.3 angstroms apart, that's a more detailed picture than if you can only distinguish things that are 5 angstroms apart. Does anyone have questions about how that works or why that is, or is that clear for everyone? So I also just had this recap slide from the slides that just shows you the difference between those kinds of resolutions. So this smallest in number resolution is small enough to show you electron density, to point out individual atoms and where they are, which can give you very detailed information about where the bonds are, where your atoms are, what kinds of shape your protein is making. And so that's smaller number because you're distinguishing smaller distances compared to like in part A on the left, a 6 angstrom resolution just kind of tells you you got a lot of electrons with a hole in the middle, which is useful but not as useful. So does anyone have questions about X-ray crystallography? All right. So this is, I believe, our last question. So let's compare techniques. So for your senior project, you wanted to determine the structure of an enzyme. Miraculously, you managed to obtain both an NMR structure and a crystal structure. However, the two don't agree perfectly. Near the active site of the enzyme, a few amino acids are in different places. One domain of the protein is rotated 15 degrees, and 10 amino acids are missing from the NMR structure. So take a few minutes and think about why might these structures be different, and what are the pros and cons of each of these structure determining techniques. you want to answer? You had a question.
I have a question.
Okay.
See rotation using NMR.
So it might tell you. So NMR is telling you which things are near each other. So if you have a rotation, some things will suddenly be close to each other, and some things will be further from each other.
Would you be able to tell us in degree?
So, I mean, not just by like looking at it, but again, it's like something that if you feed the information into a computer, the computer will use the NMR information and predict what that structure would look like in 3D. It will like the computer will draw you a picture.
Right.
Yeah. Do you have a question?
I was gonna try to answer the first question.
Okay, sure. Yeah. So why make the structures be different?
Is it because trying to put them in a crystal structure forces them into a non-native structure?
So yeah, you're definitely on the right track. So we were talking a few slides ago about how when you make that crystal structure for X-ray crystallography, you have one single conformation that all the proteins are being forced into. It's probably its native conformation. It's probably not in an unstable one, but it is one single conformation, one particular shape with very particular angles. And we talked about how that's different from NMR, where your protein is in solution. And so if you have unstructured areas of your protein in particular, you could see differences, because in NMR you would see more flexibility, especially in those relatively unstructured areas. Did you want to add to that? Or?
I was going to say it's probably mostly in the environment where, like the things that when you're using X-rays, you need them to be very structured. You know, the conformation has to be the same. They can't be. They don't actually move while NMR is in solution, which can interact with the solution as well as move freely in the solution.
Yeah, exactly. So that's why you're seeing these differences that arise. So, yeah.
I have a question about why the 10 amino acids would be missing.
Yeah.
Is it because you get like that proton spin overlapping, so therefore some protons in amino acids are not as clearly defined and therefore ultimately mixed? Does that make any sense?
So I think I see what you're saying, but I think a better way to explain why the amino acids are missing is also comes down to the movement. So if you have really fast movement of like a very unstructured area, those protons might be like moving so fast that you can't capture their spins by NMR, like by looking at that magnetic field and the decay of the spin. And so, yeah, so like that's why the difference arises. So in NMR you can't see it because it's just moving so fast. Another possible reason that something might be missing, and this might be more for an X-ray crystallography structure, but so a lot of times, especially if you have, for example, a protein that's like embedded in a membrane, it might have some regions that are in a membrane that are very hydrophobic and some regions that stick out into the environment that are more hydrophilic. And so those types of proteins can be very difficult to crystallize because the different regions of that protein prefer different conditions. And so sometimes to make an X-ray crystal, you actually have to like cut off pieces of the protein that are unhappy to be a crystal in that particular condition. So in this question, it's asking why amino acids are missing from the NMR, and that would be because of movement. But if you flipped it and said there's amino acids missing from the X-ray crystal, that might just be because you had to cut them off to get the crystal. Yeah. Yeah. So, yeah, and so for the other differences that we see, so the domain of the protein being rotated, why might we see that happening? Yeah.
Hydrophilic interactions, maybe?
So, yeah, I mean, I think for that particular question about rotation, you don't even have to get that specific. Again, it really can you can think of it coming down to just in NMR things are movement, there's more fluidity. So like maybe that one particular domain is connected to the rest of the protein by an unstructured area. And so because there's that unstructured connection, this domain can flip and flop. And so that brings us to the first part of the question, because we did this backwards. So near the active site of the enzyme, a few amino acids are in different places. So why might some of the amino acids around the active site have moved? So, yeah. So, yeah, potentially. So, I mean, you could sort of answer it by the same kind of answer we were giving for the other ones, which is there's flexibility there, which is true. But for this question in particular is asking about the active site. So, what goes into the active site? What happens there? Yeah, so this is an enzyme. It has a substrate. The substrate has to interact with the active site for the enzyme to catalyze a reaction that's happening to the substrate. And so when your substrate is bound to the active site, the enzyme might naturally change its shape a little bit because of that binding of the substrate. And so and then this comes down to sort of the practicality of it. A lot of times, especially like if you're trying to make a crystal of a protein, maybe it will only crystallize if you put its binding partner in with it. And like maybe the binding partner helps stabilize the protein near that active site. And so it's possible, we didn't state it in this question, but it's possible that in order to get your X-ray crystal, you put the binding partner in with the protein. And that gave the active site amino acids a slightly different shape or orientation compared to maybe your NMR structure, where maybe you didn't add the binding partner. Or the other way around, maybe you had to add the binding partner to do your NMR experiment, and you weren't able to crystallize your protein with the binding partner, the substrate, in there with it. And so that could be one potential reason why you'd see a difference, particularly at the active site. It's just like a practical reason. Any questions about these structure determination techniques? All right, so those are all the questions I have for Chapter 6. We have a couple more minutes, and I will leave those to you. That is my gift for your Thursday, 5 minutes of free time. And make sure for next week your paraphrasing responses, I believe, are due. And your quiz 5, which is on this chapter, is due a week from today. And if you have questions, you can come see me now or come to office hours. I'm here until 3 o'clock today. Yeah. Yeah, so the way the paraphrasing is working is so everybody posted their sort of original paraphrase, and then at this point now all of you, the students, will respond to each other and give each other feedback on the paraphrasing. At that point, I will step in and tell you this looks good, this doesn't look good, and I will let you know. I'll give you about a week before the revision is due. I'll let anyone who needs to do a revision know if they need to do a revision. Okay. The exams?